I'm Hailey YOON from the KGSEA Math Circle, and today I'll be presenting about the use of spectroscopy in biology. Spectroscopy basically is the study of how different matter interact with electromagnetic radiation. Electromagnetic radiation is a form of energy that travels in the form of both waves and photons, or particles. X-rays, UV, visible light, infrared, and radio waves are all types of electromagnetic radiation. In biology labs, spectroscopy is used to study how biological matter interacts with electromagnetic radiation. Here, biological matter indicates matter such as cells, proteins, DNA, RNA, and more. Spectroscopy is a commonly used biology lab technique due to its non-destructiveness, meaning that it does not destroy the sample being tested. Yet it is highly sensitive and can provide information on a molecular level. Therefore, it is used in fields such as pharmaceuticals, protein structure analysis, enzyme kinetics, and molecular biology. Here are some major experimental techniques that utilize spectroscopy for studying biology. These techniques all use different types of electromagnetic radiation or different principles regarding the radiation. Today, we'll be looking at the first two types of techniques. First, UV visible spectroscopy helps us measure a sample's absorbance of UV and/or visible light. The figure below shows the general process of how it's done. First, light is emitted through a light source, and a wavelength selector selects waves of specific wavelengths to go through. These waves then go through the sample, and those that are not absorbed by the sample are also detected by a detector. The signal transfers into an electric current sent to this computer for signal processing. Here we can analyze the amount of absorbance of the sample for each designated wavelength. Different biomolecules have different absorbance rates for different wavelengths, which helps scientists determine the type of biomolecule in the sample. In addition, using the Beer-Lambert's law that states that the absorbance equals the molar absorption coefficient times the molar concentration times the optical path length helps us determine the concentration of the sample. The molar absorption coefficient is designated for each certain biomolecule, and the optical path length and the absorbance are fixed values that scientists can navigate. Therefore, the molar concentration is the only value that we can calculate. This is an example of a UV visible spectroscopy result shown on a computer graph. This specific result analyzes a DNA sample. The first indicator we should look at when analyzing the results is the concentration. Typically, concentrations above 20 nanograms per microliter are considered appropriate for further analysis. As you can see here, the concentration is 65.5, which is appropriate. Next, we should look at the peak absorbance rate. Since we're looking at the DNA sample, DNA shows peak absorbance at typically 260 nanometers in wavelength. Other biomolecules, such as protein, shows peak absorbance at 280 nanometers, which helps us differentiate between the types of biomolecules. Here, the peak is generally around 260 nanometers, which tells us that the sample we're looking at indeed contains DNA. Lastly, let's look at the 260 to 280 ratio. As I mentioned before, while DNA shows peak absorbance at 260 nanometers, protein shows peak absorbance at 280 nanometers. Therefore, the 260 to 280 ratio indicates whether this DNA sample is contaminated with protein or not. Typically, for a pure DNA sample, the 260 to 280 ratio should be approximately 1.8. Here we can see that the value is 1.83, which again indicates that this is a pure DNA sample. The next technique is X-ray spectroscopy. X-ray spectroscopy uses the principle of X-ray photons. When these photons are shined through a certain atom, these photons excite inner shell electrons of atoms. When this energy is similar to the electron's binding energy to the nucleus, the electron is ejected from the atom, leaving a temporary hole that causes a sharp increase in absorption rates of X-rays. As you can see in the graph on the left, the element B shows a sharp increase in the energy between E1 and E2. This certain point is called the absorption edge. The absorption edge for each atom depends on factors such as oxidation state and coordination chemistry, which are indicators that scientists aim to analyze through these techniques. One example of how this is used is detecting DNA damage. DNA backbone contains the atom phosphorus, and when it is damaged, it changes the local environment of the certain phosphorus atoms around it. As a result, the absorption edge of the phosphorus shifts. And on the energy absorption graph, as you saw in the previous slide, new peaks appear in this graph, which tells scientists that DNA damage has been detected and also tells us the amount of DNA damage that has gone through. Other than spectroscopy itself, light and electromagnetic radiation is a tool widely used in biology. For example, fluorescence is used in fields such as fluorescence resonance energy transfer or geometry tagging, helping scientists determine the distance between molecules and further analyze the interactions between them. Transmission electron microscopy, or TEM, utilizes electron beams that are essentially electromagnetic radiation with extremely short wavelengths to visualize stereoscope of cell organelles, which cannot be detected with normal light microscopes. In conclusion, light and electromagnetic radiation is a valuable tool in the realm of biology and shows that advanced experimental techniques can aid in recent science breakthroughs. Thank you.